So, Mr. Chairman, with your permission, I will move on. Okay. So, what I'd now like to talk to you about are some new and emerging agents for nuclear medicine molecular brain imaging. And these will cover a spectrum of 2 or 3 focal areas, but I think you'll see areas where progress is being made and where new tracers, certainly in the research arena, are becoming available. And I think some or all of these will end with molecular approaches that will be available to us for clinical diagnostics within the coming few years. So here's the outline of what I'd like to talk to you about. First, new vesicular neurotransmitter transporters. These will be targeting the VMAT2, which we've already talked about, but there's a new tracer out there that I think has the possibility for clinical translation. And then a new tracer for the vesicular acetylcholine transporter. Based on the disappointing news I've just shared with you about the performance of PK11195, you will be happy to learn that there are new radioligands of the translocator protein that are under development, and these show great promise, I think. And then finally, with regards to the imaging of amyloid aggregate depositions, you know about A-beta amyloid and its role in Alzheimer's disease, and that there are 4 agents that are now ready for clinical translational use, and hopefully 1 or more of these will become available to us for clinical use. But there are other amyloid aggregations, and particularly of the tau protein, where there are some very interesting ligand approaches that have great promise, and I'll share with you some of the preclinical and, in one case, clinical evidence for this. So to start with the vesicular transporters, we've talked at some length about VMAT2 and imaging based on tetrabenazine derivatives, particularly dihydrotetrabenazine. Investigators at Pennsylvania University, Hank Kung and his colleagues at Avid Radiopharmaceuticals have developed a derivative of dihydrotetrabenazine with fluoroalkylation of this position. So we now have fluoropropyl dihydrotetrabenazine, which Avid Radiopharmaceuticals has badged AV133. Now, we thought that placing this kind of a bulky substituent at this location on the molecule might result in completely inactivating its pharmacologic activity. We were completely wrong. The affinity of AV133 for the VMAT2 site is actually 10 times higher than the parent dihydrotetrabenazine. These are representative images as a function of time. So what we have here in each column is a rostral to caudal transaxial image, and this is 0 to 2 minutes, 15 to 20, 60 to 90, and so on, out to 200 minutes in a normal individual. And what you can see is that AV133 enters the brain readily. It shows selective retention in the striatum, and this selective retention persists for hours after its injection. This can be analyzed kinetically together with blood samples for the authentic tracer, and we can come up with these K1 and volume of distribution binding parameters, which basically extract the features out of this time series that relate to initial delivery of the tracer versus its retention. And you can see this volume of distribution map compares very favorably to what I was showing you earlier with carbon-11 dihydrotetrabenazine. But even better than this is the apparent distinction in Parkinson disease. So here's a normal control, and what you see is intense binding in the striatum, and these are parametric volume of distribution images. If you look at this individual single subject, though, you can actually see the brainstem, you can see the substantia nigra on a single subject basis because the fluorine 18 has such superior imaging characteristics. These are 5 individuals with mild idiopathic Parkinson disease, and you can see a range from least severe to most severe across these. But again, in each of them, you can see an abnormality of the binding in the brainstem as well. This is a single individual that we studied in who has dementia with Lewy bodies, and note that he has a bilateral and very severe lesion, even compared to almost the most severe of these PDs. So AV133 looks like it has very favorable imaging characteristics, and because of its fluorine 18 label, it can be synthesized regionally in cyclotron facilities and distributed to a variety of nearby hospitals for administration and imaging, just like F18 FDG. Now, this is not yet approved by the FDA for use in the U.S. Avid Pharmaceuticals holds the patent license for it, and I do believe they plan to pursue registration and licensure in the future. We've looked at the ability to discriminate Parkinson disease from controls, and it turns out that with our parametric measures that we would favor, in this case measuring the relative volume of distribution, it's actually no better at distinguishing patients from controls than the simple pattern of radioactivity at 2 hours or so after injection. So this looks very favorable then for clinical use, where a simplified imaging approach to inject, wait 2 hours, and image for perhaps 20 to 40 minutes would suffice and give a very significant non-overlapping pattern in Parkinson's disease compared to normal. Now, the second vesicular transporter, where there's nice underlying neurochemistry as well as radiochemistry, is the acetylcholine vesicular neurotransmitter transporter. So this is a measure then of presynaptic cholinergic terminals, and these in turn are of great interest in the degenerative dementias, where the cholinergic cells in the base of the forebrain are known to degenerate, both in dementia with Lewy bodies and Parkinson's disease with dementia, as well as in Alzheimer's disease. And one of the pressing questions is, what is the role of these neurons in the evolution of cognitive decline and dementia? So this kind of an approach would allow us to address that question. The ligand that forms the basis for these ideas is called visamicol, that's this structure, and fluoroethyl benzovisamicol, with this guy tacked on the end, allows us to radiolabel with fluorine 18. Now, we've been pursuing this for many years in our laboratory because visamicol itself is very neurotoxic. It has a curare-like neuromuscular paralyzing effect because as it blocks acetylcholine transporters into the synaptic vesicle, it can cause a failure of synaptic transmission at the neuromuscular junction. FeOBV, on the other hand, can be synthesized at high enough specific activity that it does not have significant pharmacological effect associated with it. And we've just this month reported our initial experience with this agent. So here's a series of brain imaging acquisitions in a normal volunteer over time, showing that in the basal ganglia, where there's a very high concentration of intrinsic cholinergic interneurons, the agent continues to accumulate for many hours after injection, whereas in brain regions that have less, or in the case of the cerebellum, virtually no presynaptic cholinergic innervation, the time course is much more reversible and amenable to kinetic modeling. These are brain images. First, this is the volume of distribution parameter from that 4 hours of imaging, showing again intense accumulation within the striatum, and this is because of striatal interneurons that are cholinergic. Here is the patient or the subject's MRI, and here's the fusion. If we scale up this pattern by a factor of about 5, you can see that there is actually detectable signal, readily detectable signal, throughout the cerebral cortex. This is on the basis of basal forebrain innervation. And there's also considerable signal present in the thalamus, and these presynaptic cholinergic terminals arise in the brainstem. So there's actually another cholinergic projection, brainstem to thalamus, that can be imaged and quantified with this approach. But again, to make these measures requires hours of imaging in each subject, and you can see that there's acceptable precision in the cerebral cortex and in the thalamus, but in the basal ganglia, in the striatum, there's still about a 25% error with estimation of binding, and this degree of variability gives us some concern that we may not be able to image and quantify a striatal binding as accurately as we might like. If we throw away the kinetic modeling aspect and simply look, though, at the late pattern of tracer retention, this is the volume of distribution map, and this is simply the late pattern of radiotracer retention, and they become very, very similar looking at 3 to 4 hours after injection, which suggests that we may be able to approximate the binding by simply a late distribution. And here again is the same, but scaled up by a factor of 5-fold, so you can better see the distribution of lower intensity binding here with parametric calculation of the binding potential, and here simply with the late retention. So we think this radioligand has great promise for facilitating studies of the acetylcholine transporter, and will give us a second target on yet another vulnerable neuron population that is of great interest in both Parkinsonian and dementia neurodegenerations. So let me turn now to TSPO. So this is that nice target on mitochondria in activated astrocytes, white blood cells, and so on, where PK1195 was not a very well-behaved radiotracer. And you remember these were the limitations we identified with its use. Well, it turns out that there's a wide range of chemical structures that have now been discovered that interact with the TSPO at very, very high affinity. And this paper I refer you to in the European Journal of Nuclear Medicine, for instance, articulates many classes of chemical structures that now are known to interact at high affinity with the TSPO. And you may imagine then that these chemical families give rise to the possibility of many radiolabeled tracers. One of these that's probably most extensively studied up until now is carbon-11 labeled PBR28, so that's peripheral benzodiazepine receptor 28. And this was introduced by Vic Pike as a radiochemist and Bob Innes at the National Institute of Mental Health in the US. And they identified this amongst related chemical structures as a tracer that interacts with very, very high affinity with the TSPO protein. And indeed, when injected into man, it has a biodistribution that's very favorable. So with initial injection, it spreads predominantly to peripheral organs, but you can see brain activity and uptake, which is very faint but present. But as time progresses, you can see that it shows retention in the spleen, and we think that's because of the known binding to spleen and here in the kidneys because of the TSPO protein in those areas. The liver activity, on the other hand, we think is because of metabolism of the ligand, and in late studies you can see accumulation in the gallbladder, which reinforces that idea. A very interesting anomalous pattern was seen in several patients that these investigators initially reported, and that is the initial biodistribution looked just like this normal individual, but that delayed retention studies did not show evidence of activity in the spleen or in the kidneys, but did show metabolism of the tracer and excretion in bile and in the urinary bladder. This pattern is referred to by these investigators as that of a non-binder, and they find this is present as much as 20% of the time when selecting normal volunteers for study. It is exactly what one finds in experimental animals when you compare the biodistribution of PBR28 in an untreated animal with the biodistribution you get by giving a previous blocking dose of non-radioactive PK11195, which makes this retention study look just like a human non-binder. What we now know from more intensive study of binders versus non-binders is that there's a single base polymorphism in the gene responsible for the TSPO binding site, and those with this polymorphism express a very low affinity for these new tracers and hence their inability to be detected in vivo. There are several different ligands that have been discovered as son of PK11195, and they all display this distinction between individuals who are high affinity binders versus those that are so-called non-binders who are low affinity. And you may imagine, and you would be correct, that there are also mixed binders who have one of these alleles and one of these. All of these newer agents, though, have a very high affinity for the TSPO site. They all have been selected for very good blood-brain barrier penetration, and they're amenable to simple quantification with binding ratios. And here's just a teaser example from what the Innis examination, Innis Laboratory has published in their initial examination. So this is a normal volunteer that was recruited to their study, and here's his normal MRI. But this is his PBR28 binding map obtained a week later. And what they found and could not readily explain is that there's this intense focus of increased binding here that they would not have predicted or could not understand. They immediately brought the patient back for repeat MRI, and in the week between this baseline study and the PBR scan, he had developed a lacunar stroke here in the internal capsule region. So this shows the ability to image the inflammation associated with a subacute stroke that was unfortunate for their experimental design, but very informative about the possibility of this agent showing us relevant neuroinflammation changes. So watch more for this ligand. In research labeled with carbon-11, there is a fluorine-18 analog called PBR06, which shares the same chemical backbone and structure, but is obviously labeled with F18 and may be amenable then to regional distribution for clinical use. I'd like to close then by talking about amyloid imaging, and this implies amyloid like A-beta plaque amyloid, but also the amyloid associated with tau protein aggregation. So we talked yesterday about the notion that A-beta amyloid is characteristic of a number of disorders, including Alzheimer's disease, some patients with dementia with Lewy bodies, and also those with amyloid angiopathy. But there are a host of neurodegenerations where tau accumulations are the amyloid of interest. And we don't really have, at the present time, anything that would help us to identify the tau accumulations in progressive supranuclear palsy, frontotemporal dementias, or corticobasal degeneration. So this now is an area of potential interest. I did mention to you yesterday there are 3 fluorine-18 A-beta amyloid avid tracers, and hopefully these will become available for clinical use in the near future. 3 of these are approved, florbetadine in Europe and flutometamol and florbetapir in the United States. The Navidea compound, 4694, to my knowledge is not yet approved, but is undergoing the same evaluation pathway as these other agents. So, again, to return now to emerging interest in finding similar compounds that would identify the tau depositions in these conditions, the first thing we need to do is to step back and say, well, tau, is that as simple as A-beta amyloid? Everybody's A-beta amyloid has the same molecular structure. The A-beta peptide only comes in a 2-peptide length disparity, 1 to 40, 1 to 42, and all A-beta amyloid plaques in Alzheimer's disease patients have a mixture of those 2 peptides, and the ultrastructure of the amyloid is all the same. So there's a plausible target that these amyloid imaging agents that have shown success should be looking for. Tau, on the other hand, is a much more complicated target. First, tau is not a single protein in the normal human brain. There are at least 5, and perhaps a 6th, although this is found most robustly in fetal brain, but there are at least 5 isoforms of tau that are different one from another by the expression of different of these variable exons that are either included or excluded by splicing of the mRNA. Tau has fundamentally two families. One group of taus are referred to as the 4-repeat or 4R tau types, where there are 4 of this one epitope that's in the microtubule binding region. So I should have told you the tau protein is a microtubule-associated protein whose binding to tubulin stabilizes microtubules in the intracellular neurons. So there are 4R taus with 4 of these repeat domains that are responsible for binding to tubulin. And then there are the 3R taus that lack exon 10, this one, it's been spliced out, and so they have only 3 of these repeat epitopes. So these are fundamentally different proteins at the primary structure level, and then there are some variable expressions of exon 2 and 3 amongst these as well. So what this does is it gives rise to the possibility of many different primary tau protein structures that might accumulate pathologically in different diseases. And you would have been right to speculate on this, so that the tau that's part of neurofibrillary tangles in Alzheimer's disease includes both 3 and 4 repeat primary structures. It's located in neurons, of course, so these are the neurofibrillary tangles. And at an ultrastructural level, these are highly cross-linked, very tightly bound structures. So these are the neurofibrillary tangles in Alzheimer's disease. But tau is also deposited in a more amorphous form in Pick disease, one of the frontotemporal dementias. Here, though, the tau primary structure is entirely the 4-repeat length form. There's no 3R. It accumulates in neurons as a kind of fluffy intracellular accumulation. And here, it's present as straight filaments and random coiled filaments, but none of this highly cross-linked fibrillary appearance. So it's different in primary structure, but also in its tertiary and aggregate structure. Now, in progressive supranuclear palsy and corticobasal degeneration, so those movement disorder-related tauopathies, the isoform that's involved in the deposition here is the 3R form. And it's not in neurons, it's in astrocytes in these diseases. And here it's very loosely affiliated as straight filaments and or twisted filaments, which are much more amorphous than the neurofibrillary tangles. So whether we talk about the primary structure, the ultrastructure of the tau accumulations, and or the cells in which we find the deposition, there's great variability across a range of neurodegenerations that we would be interested in. So in the development of ligands that target these tau depositions, one has to be mindful that there are multiple targets here associated with primary and post-translational processing changes and variability in the aggregate structure. So anytime someone finds a ligand that they say binds to tau or to tau aggregates, we need to ask, well, what kind of tau aggregate was that? And what is the range of sensitivity and specificity of the binding? So if we have a new tracer, is it really specific for tau, or are there other peptide aggregates that it might react with, particularly A beta amyloid, for instance? Is it specific for one or more of the tau isoforms? And is it specific for one or more of the aggregate structures? And these are all questions that would need to be answered for any and each individual tau radiotracer that might be proposed. And I'll show you, there are several that show evidence of possible success. So this is one from our laboratory, N-methyl lansazoprol. It has this structure, and we find that in vitro it binds to tau in progressive supranuclear palsy samples, so it's at least finding more than a simple parahelical filaments in Alzheimer's disease. It enters the normal monkey brain readily and has very favorable kinetics, but we have not yet imaged human subjects with neurodegeneration with this agent yet. There are other structures from other laboratories. So here, from the laboratory of Zhang et al. Is a compound T808, which is able to be labeled with radiofluorine. And what they've shown in vitro, at least, is that it's able to bind to tau depositions in a variety of neurodegenerative cases, some of which are Alzheimer's disease and have lots of A beta, and others of which, though, are relatively A beta absent and presumably FTD or related. And in these conditions, there is persistent binding of T808, suggesting that it may indeed be recognizing tau as opposed to A beta. Yet other investigators here from Australia have taken THK523, a structure somewhat reminiscent of T808, and found in vitro that the immunochemical deposition of tau protein in the temporal lobe of patients with Alzheimer's disease looks very pattern similar to the binding of this agent, which looks in turn very different from the A beta amyloid staining in the same sections. They have gone on to inject this radiotracer into transgenic mice. This is a wild-type mouse showing not very significant retention in the brain. Here's a transgenic mouse with a tau mutation showing increased brain uptake. And here is a transgenic mouse with a amyloid precursor protein presenilin mutation that leads to the deposition of A beta amyloid, showing relatively less accumulation than here with the tau mutation. So at least in the transgenic mouse, this agent appears to have nice selectivity for tau protein aggregates over A beta amyloid. To my knowledge, it's not been used yet in humans. And then there's this very exciting compound from a Japanese group published about a year and a half ago in Neuron. This is the baseline structure for the compounds that they've investigated. In this one, they designate PBR3 as one that has the most attractive in vitro appearance. This has been studied in humans, and this is its distribution in a patient with a typical Alzheimer's disease. So here is a PIB binding to confirm that there's amyloid deposition, and here is the distribution of PBB3 in a coronal section. Notice that in the Alzheimer's patient, there's not a lot of amyloid here in the hippocampal formation, whereas there's selective and very intense binding of PBB3 because of the known neurofibrillary degeneration in this location. So this is evidence of binding in probably to neurofibrillary tangles in typical Alzheimer's disease. But this is even more exciting. So this is the distribution of PBB3 in a patient with corticobasal degeneration. So this is the 3R amorphous tau accumulation that's seen in astrocytes. And what they find is that in the thalamus and basal ganglia, there is intense accumulation of PBB3, which suggests to us that it is indeed recognizing this very distinct tau from the neurofibrillary tangles that it also appears to identify. And this individual is, of course, PIB negative because they don't have Alzheimer's disease. So to summarize where we've come and where we may be going, we have a number of tools available already to us for the molecular endophenotyping of degenerations and dementia. FDG is a very powerful tool, as we talked yesterday, and we'll talk again at the end of the day today. We have a number of Fluorine 18 labeled probes for fibrillary A beta amyloid deposition, many of which should be available for our clinical use. I believe we'll soon have fluoropropyl DTBZ, the AV133, but we already have DAT scan and 18F fluorodopa to look at the dopamine neuron projections. What we really want, though, is a way to confirm that an amyloid-negative neurodegenerative case is indeed related to tau deposition, as in, say, Pick disease and some other frontotemporal dementias, versus other proteins that accumulate in some frontotemporal dementias. And in fact, TDP43, a non-tau-related product, may be the second most common protein accumulation in frontotemporal dementias. Some think it's even more common than tau accumulation. So we would really like to have ligands that target not only tau specifically, but perhaps also progranulin, TDP43, and C9orf as additional peptides that accumulate in different types of FTDs. With regards to neurodegenerative movement disorder phenotyping, we already have nice ligands for the nigrostriatal projection, and we've talked at length about these. What we would really like to have, though, would be a tracer that might show us alpha-synuclein deposits, characteristic of Parkinson disease and multiple systems atrophy, versus the 3R tau that would tell us the patient has PSP or corticobasal degeneration with some degree of certainty. And as you've seen, there is an agent already on the menu that may be able to do this by example of that single case of corticobasal. We do not yet have a clear candidate for alpha-synuclein aggregate imaging, but would really like to have one. So the other thing, and I'm not going to spend any more time other than this to talk about it, what we talked about just on the last 2 slides was molecular endophenotyping, that is classifying what kind of degeneration a given individual has. But the other thing that we would like to have to more rapidly detect effective therapies in our clinics would be to have markers that tell us about whether the disease is progressing or whether we've had a significant ability to halt or slow the progression. And what would we use for measuring that aspect? Well, we could possibly use FDG as evidence that it progressively worsens in Alzheimer's disease. With regards to nigrostriatal dopamine lesions, we can use VMAT2 perhaps. With regards to basal forebrain cholinergic neurons, we could theoretically use the vesicular acetylcholine transporter. But notably absent here, what we don't have is a good objective measure of neuronal survival versus further degeneration that's a specific marker for glutamate or GABA neurons, and these are the vast majority of nerve cells in the brain. And we think it's glutamate neurons in particular that in many of these neurodegenerations that are the target of symptomatic progression. So we'd really like to have some nice objective measures of these nerve cells that we could use as an independent indicator of disease progression and or the effect of new therapies on the progressive disorder. So this just summarizes what we've just talked about, and I think to allow time for discussion, I'll conclude here.